Hi, my name is Lauren Weiss,
Hi, my name is Xiyu Xiao.
and we're in MCDB 427. Today we're going to talk to you about Figure 6.42 in our book, Molecular Biology, by Robert F. Weaver. Transcription can be terminated in two different ways. Here we will focus on a model for intrinsic termination. It involves a hairpin loop and uracil base pairing. In this model, RNA polymerase pauses. This pause grants a previously transcribed portion of the RNA the opportunity to fold in on itself and base pair, forming a hairpin loop. Note that this base pairing will only occur if the RNA contains an inverted repeat. The formation of this hairpin destabilizes the RNA-DNA hybrid. There exist two hypothesized mechanisms of destabilization. One, that the formation of a hairpin loop actually pulls the RNA out of the RNA polymerase, and two, that the hairpin causes the transcription bubble to collapse and release the RNA from the DNA-RNA polymerase complex. The question that Figure 6.42 in our book responds to is, how do you know that a hairpin loop is important in transcription termination at all? W. S. Yarnell and Jeffrey Roberts performed an experiment in 1999 hoping to find an answer to this question. In this experiment, they performed a runoff transcription assay. Runoff transcription assays are performed in vitro. RNA polymerase is allowed to transcribe the template DNA of known length so that the transcript produced is also of known length. There is no termination site on the DNA, so the RNA polymerase simply runs off the template, hence the name of the assay. Transcription proceeds in the presence of radiolabeled nucleotides, so the transcripts produced can be electrophoresed and autoradiographed for visualization purposes. The thickness of the band corresponds to the amount of transcription, and the location of the band on the gel corresponds to the transcript length. The DNA template used by Yarnell and Roberts contained two mutant terminators named delta TR2 and delta T82 respectively. Each mutant terminator sequence contained a stretch of T nucleotides on the sense strand, so a stretch of A nucleotides on the template strand, but was missing half of the inverted repeat needed to form a hairpin loop. Under these conditions, no hairpin would form. To produce both of these mutant terminators, the experimenters removed half of the terminator sequence from the DNA. The arrows above the DNA sequence denote the remaining portions of the terminator sequences, respectively. Yarnell and Roberts coupled the DNA templates used in the runoff assay to magnetic beads. This coupling was possible through the attachment of biotin to the DNA, which is in turn attached to the streptavidin bound to the magnetic bead. One more note before we introduce the image of the gel that Yarnell and Roberts produced. Any radiolabeled mRNA that did not terminate will remain attached to the DNA template and therefore to the magnetic bead. This is because termination releases the transcript from the template. Without termination, there will be no mRNA release. The RNA polymerase might pause at the stretch of T nucleotides leading up to their respective mutated terminator sequences, but the transcript will not be released without a functional terminator. After centrifugation, paused transcripts will wind up in the pellet. Ultimately, however, the RNA polymerase will continue transcribing through those mutated sequences after pausing, since there is no functional terminator at these sites. The polymerase will reach the end of the DNA template and run off, releasing a full-length transcript. After centrifugation, the transcript will wind up in the supernatant. Additionally, any radiolabeled mRNA that did terminate will be released from the template DNA and therefore from the bead and will wind up in the supernatant. This is a huge gel. We'll walk through the lanes in groups. First, let's take a look at lanes 1 through 6. Here, the DNA template, which we saw in part A of the figure, is incubated with RNA polymerase and labeled nucleotides. Lanes 1, 3, and 5 are labeled S, to denote that these electrophorese products were found in the supernatant, and lanes 2, 4, and 6 are labeled P, to denote that these electrophorese products were found in the pellet sample. We also see a label beneath the lanes indicating the lack of oligonucleotide in solution. We'll explain the role of the oligo later, just keep in mind that it's absent in lanes 1 through 6. Over the course of 600 seconds, full-length mRNA accumulates in the supernatant. This band is labeled runoff, since it's the full-length product of runoff transcription. On the gel, we see that the amount of full-length mRNA increases with time. There are two other bands in lanes 1 to 6. And these correspond to delta TR2 and delta T82 terminated transcripts in the pellet sample respectively. We see that these bands disappear over the time course. The RNA polymerase pauses at these mutant terminator sequences due to the respective strings of adenine nucleotides on the template strand, but it does not release transcripts at either mutant terminator as we described earlier. Instead, these transcripts become full length and are released into the supernatant when the RNA polymerase runs off the template DNA. That's why the total amount of runoff increases over the time course. Per the gel, it just takes a little while for the transcript to become full length. Ultimately, you would expect only full-length mRNA to be present in the supernatant under these conditions. Now let's look at lanes 7 through 9 and then 10 through 12. Here the experimenters have added a T19 or a T18 oligonucleotide. The T19 oligonucleotide is complementary to the stretch of underlined DNA depicted here. Note also that the T18 oligonucleotide is complementary to the DNA depicted there. These oligonucleotides bind the mRNA and allow for the formation of structures similar to hairpin loops at their respective locations. Yarnell and Roberts postulated that the mechanism of termination with an oligonucleotide would look something like this. Instead of the formation of a hairpin within the transcript itself, the oligo would hybridize to the transcript, creating a double-stranded molecule. This molecule parallels the double-stranded nature of a hairpin loop and would induce termination. Note that these oligo-induced mutant terminators are less efficient than wild-type terminators. RNA polymerase pauses at these sequences, but true termination does not often occur. Back to the gel. We'll focus on lanes 7, 8, and 9. Here, the T19 oligonucleotide was introduced in the solution, and transcription was allowed to occur. Notice that all of the electrophoresis products in these 3 lanes come from the supernatant. As we discussed earlier, mRNA samples in the supernatant experienced termination and were released from the DNA template. We see a heavy banding pattern at the delta TR2 site. The banding pattern at the delta T82 site is essentially unchanged. Additionally, the runoff banding pattern increased in intensity over the 600-second time course, but this is less total runoff than in lanes 1 through 6. The T19 oligonucleotide that was introduced for this portion of the assay must be increasing termination at the delta TR2 sequence, since there is more delta TR2 length product in the supernatant. These products were released from the template DNA. Termination of transcription is what allows the release of non-full length transcript. Because of this transcript release, there is less full-length mRNA produced. Hence, there is less total runoff than in lanes 1, 3, and 5. We can attribute the delta TR2 termination to base pairing between the T19 oligo and the mRNA, since the addition of oligo to solution is the only change between lanes 1, 3, and 5, and lanes 7 through 9. Somehow this oligo causes termination. This means that an intramolecular hairpin is not necessary for termination to occur, but some base pairing event is. Now on to lanes 10 through 12. In this case, the T18 oligo was introduced into solution and transcription was allowed to occur. Notice again that all of the electrophoresis products in these 3 lanes come from the supernatant. So these are products that were released from the DNA template. The banding pattern at the delta TR2 site is essentially unchanged as compared to the delta TR2 banding pattern in the absence of the T18 oligo. We see a heavy banding pattern at the delta T82 site. Additionally, the runoff banding pattern increases in intensity over the 600-second time course, but there is less total runoff than in lanes 1 through 6. The T18 oligonucleotide that was introduced for this portion of the assay must be increasing termination at the delta T82 sequence since there is more delta T82 length product in the supernatant. These products were released from the template DNA due to termination, causing less full-length mRNA to be produced than in lanes 1, 3, and 5. We can attribute the delta T82 termination to base pairing between the T18 oligo and the mRNA, since the addition of the oligo to solution is the only change between lanes 1 through 6 and lanes 10 through 12. We can reach the same conclusion as we did for lanes 7 through 9, which is that an intramolecular hairpin is not necessary for termination to occur, but some base pairing event is. And another takeaway here is that reconstructed TR2 is a better terminator than reconstructed T82. How do we know this? Well, if there are darker bands, then there are more transcripts of whatever length of mRNA corresponds to that band. The banding in lanes 7 through 9 at the delta TR2 site is darker than the banding in lanes 10 through 12 at the delta T82 site. So more transcripts are terminated at the TR2 terminator sequence than the delta T82 terminator sequence in the presence of an oligo. Why is this? Well, the question is, what factors influence how good a terminator is? Perhaps the GC content, the spread of GC content along the terminator sequence, and the length of the T stretch on the sense unit of the DNA. Now, these are not things that we'll focus on here, but it's an interesting side note in regards to this figure. Now let's look at lanes 13 and 14. Here the experimenters have added a new oligonucleotide, T19H1. The experimenters produced a point mutation in the T19 oligo at the nucleotide labeled with an asterisk. Other than this point mutation, the T19H1 oligo has the same sequence as the T19 oligo. Now, this mutation should interrupt base pairing between the mRNA transcript and the oligo in solution. Based on the gel, disruption does seem to have occurred. In lane 13, the DNA sequence depicted in figure 6.42a is transcribed, and the transcript is allowed to hybridize to the T19H1 oligonucleotide. The sample here is from the supernatant at the 600-second mark of the time course. Any products we see must have been released from the DNA template. So we see a much lighter band at the delta TR2 site than we saw in lane 9 at the 600-second mark of the time course. So remember that in lane 9, the oligo added to solution is exactly complementary to a stretch of nucleotides in the transcript. We can conclude from this data that termination at the delta TR2 sequence was greatly reduced by the point mutation in the complementary oligo, T19H1. However, when we compare lane 13 with lane 5, we see that termination at the delta TR2 site was still better in the presence of the mutated oligo than in the total absence of one. So we can conclude that partial complementarity enhances termination to some extent. In lane 14, the experimenters made a compensatory mutation in the DNA template to allow uninterrupted base pairing of the transcript with the T19H1 oligo. With complementarity between the oligo and the transcript restored, a double-stranded molecule was rescued. And we can conclude here that the restoration of a double-stranded molecule restores transcription termination, as we can tell by this band, and can do so sufficiently even though it is not a hairpin but rather an RNA-DNA hybrid. Finally, we'll look at lanes 15 and 16. We're in the home stretch. Now, in this case, transcription was allowed to proceed in the absence of any oligos on the mutant DNA sequence, which we originally saw as our compensatory mutant in lane 14. I'll mention now that this compensatory mutant template strand also has a wild-type T82 terminator as part of its sequence. That's why in lane 15 we see a dark banding pattern at the T82 site in the supernatant sample. These wild-type T82 transcripts were efficiently terminated and then released from the DNA. We also see that the delta TR2 banding patterns in lanes 15 and 16 match the banding patterns in lanes 5 and 6. So, in both cases, no delta TR2 length transcript is terminated and released into the supernatant, although pausing does occur at this site, as we see in the pellet sample. So these last two lanes serve as a control. They show that in the absence of a complementary oligo, namely T19H1, the termination will not occur at the delta TR2 terminator, but it will occur at this wild-type T82 terminator. So overall, this experiment by Yarnell and Roberts shows that an intramolecular hairpin loop or some intramolecular base pairing event with the transcript is important for termination. And we hope that this video helped you understand this very complex figure and that you enjoyed watching it.